Assalamu alaikum, all of you. I am Maria Maqbool from Government College University, Lahore. The title of my research is Protective Potential of Mentha piperita Extracts on Rotenone-Induced Motor Deficits in Mice. First of all, I will briefly explain about motor deficits. Motor deficits or dysfunctions are particularly defined as loss or damage of uncontrolled and coordinated movement in a person's body. These motor dysfunctions or deficits are strongly related with the different neurodegenerative diseases as they are serious issues. These issues are related with Parkinson disease, as Parkinson disease involves the loss of coordinated and controlled movement in the body. Motor deficits are caused by several environmental as well as genetic factors, but the experimental animal models can also be generated with the help of the rotenone. Rotenone is a neurotoxin, plant-based neurotoxin that induces motor deficits in an animal model. This was firstly used for research purpose in 1980s. This is the pictorial diagram uh, of a person who was suffering from motor deficits. In this diagram, we can see that there are tremors in the hands and legs of the person, whereas the elbows and the knees of the person are flexed, and the person's back is rigid, and he cannot maintain its straight body posture, as well as he is unable to walk in a proper way. Next, Mentha piperita. I have used a Mentha piperita for my research purpose in order to check its protective role against the motor deficits. Mentha piperita is commonly known as peppermint, whereas it is commonly known as pudina in Pakistan. It is an aromatic perennial herb with deep green colored leaves and light purplish or pinkish flowers on it. It is used in several synthetic products due to the presence of menthol in it. Its decoction is widely used as a drink. It has several medicinal properties, such as it has antioxidant activity, antimicrobial activity, anti-cancer, neuroprotective, anti-inflammatory, hepatoprotective, and also have anti-allergic activity. Aims and objectives of my study was to investigate the protective effect of Mentha piperita through the preparation of methanolic Mentha piperita leaves and root extract, whereas the development of rotenone-induced motor deficits in mice model, as well as the assessment of motor functional deficits in Mentha piperita treated as well as in untreated mice. The methodology of my research work was firstly include the preparation of Mentha piperita leaves and root extract. For this purpose, I dried the leaf and root of the Mentha piperita plant separately and then ground them into a fine powder. After this, the powder was mixed with methanol in the ratio of 1:10, and the flask was placed on flask orbital shaker for the period of about 24 hours. After that, the solution is filtered and the filtrate is placed on the water bath at 42 degrees Celsius for about 12 hours. After that, I lyophilize the Mentha piperita roots and leaf extracts. Next one is the experimental design and grouping of animals. My experiment consisted of total 28 days, in which I firstly treat the animals with the animals include the pre-treatment, in which I treat the animals with Mentha piperita extract without the administration of rotenone, while the next 21 days included the post-treatment with Mentha piperita extract along with the administration of rotenone. I took a total 30 male albino mice and divided them into 5 groups. The first one is the control group, which was only given 2.5% DMSO dissolved in sunflower oil at the dose of 10 ml per kg. It was injected subcutaneously. Next one is the rotenone group. Rotenone group was given 2.5%, 2.5 mg per kg dose of rotenone Rotenone was dissolved in 2.5% DMSO and 1 ml sunflower oil. Rotenone was also injected as subcutaneously. The other three treatment groups, that is the cinnamate plus rotenone group and the methanolic Mentha piperita leaves and the root groups, were also given rotenone subcutaneously after 1 hour of the oral dosing. The cinnamate group was given cinnamate at the dose of 20 mg per kg orally, while as the mentha piperita leaves and root extract were given the extracts at 200 mg per kg orally. And all these groups were given while also given rotenone subcutaneously. The next are the behavioral assessment. After this, I assess the animals behaviorally. For this purpose, I perform different behavioral tests. The first one include the narrow beam walk test. This is the apparatus of narrow beam walk, which was consisted of a wooden rod, and the length of the rod is 1.5 meter, and it is 10 millimeter in diameter. This wooden rod is placed at 60 centimeter height, and it was placed the both the stands was placed at the two ends of the rod. The animals were taken from their cage and placed at one end of the rod and allowed to cover the distance of 1 meter under the stress. After this, the time was noticed. All the activities of the animals were recorded with the help of the video camera, and two trials were given to those animals. Two trials were given to every mice. After this, their average reading was taken. This is the graph which shows the time taken by different groups. This shows that the control group took the average time of about 9 seconds, which was the lesser time as compared to the other groups. While if we look at the treatment groups, that is cinnamate, rotenone, as well as cinnamate leaf extract and the root extract group, these shows the time which is nearly equal to the control group. While if we look at the rotenone group, this group took the average time of about 35 seconds, which is the time greater than the other treatment as well as the control group. This type of behavior shows the severe motor deficits, while the treatment group shows the recovered symptoms. The next one is the pole climb down test. Pole climb down, this is the apparatus of the pole climb down test. It was performed in order to show the stiffness and slowness in the movement of animals. Animals were placed at the top of their pole and then they are allowed to descend from the pole. Their activities were recorded with the help of the video camera and the time taken to descend from the pole was noticed. This is the graph which shows the time to descend from the pole. The control group was given 9 seconds, while the treatment groups were shown the 11, 12 and 13 seconds. This is the average time. All these time were very close to each other as well as the control group. While if we look at the rotenone group, the rotenone group took the average time of about 25 seconds, which shows that this group shows the severe form of motor deficits, while the other treatment group shows the recovered motor symptoms. The next is the tail suspension test. Tail suspension test was performed in order to check the immobility time of the animals. For this purpose, the animals were hanged at the wooden apparatus, which consisted of 4 chambers of equal size. The animals were hanged for about 6 minutes and their activities were recorded with the help of the video camera. After this, the immobility time of the animals were recorded and the graph was plotted. This is the graph which shows the immobility time of different groups. Control group shows the 90 seconds, whereas the cinnamate as well as the leaf extract group shows the lesser immobility time. Whereas root extract show a little deviation and it shows the average time of about 150 seconds. But this is lesser than the rotenone group. Rotenone group, the average showed the average time of about 230 or 240 seconds. This type of behavior shows the severe motor deficits. Next test is open field test. Open field test was performed in order to check the exploratory rate of the animals when they allowed to move in an open field under no stress. This apparatus consists of a wooden chamber of equal, which is of equal sides, and all the activities of the animals were recorded with the help of the video camera. This is the time which shows. This is the graph which shows the activity of the animals and number of entries in an open field. Because the open field apparatus consists of 16 small squares, this is the number of entry in each small square. Shows that the rotenone group shows the lesser number of entries as well as the other treatment groups. This shows that the rotenone group shows the motor deficits. These are the trajectories. The trajectories of the mice were also drawn in an open field. This is the trajectory of the control group, which shows that the mice covered the greater distance when it allowed to move in an open field, while the rotenone group showed the lesser distance covered, as well as it shows that the mouse only moved along the boundaries of the apparatus. This type of behavior showed motor deficits, while the cinnamate, leaf extract, and root extract group show the greater distance covered by the animals. This type of behavior showed the recovered motor symptoms. Next is the conclusion. Conclusion of my research was that motor deficits were observed in a rotenone group, and oral dosing of Mentha piperita extracts recovered the motor impairments in treatment groups, whereas Mentha piperita can also be considered as potential therapeutics for further investigations. Thanks to all of you.